First of all, thank you very much, Enrico, for inviting me here today. I hope that your genome center will be a real add-on to the Italian community for sequencing very different kind of organisms and stuff. I believe that the application, as my colleague before said, the application to clinics will be one of the key applications for next generation sequencing in the future. We are really moving close to the patient in order to find out which is the best therapy for several diseases. So it's a real goal, I think, also for the companies that are selling this kind of machine to move directly to medicine and to the personalized medicine. Today I will present something different because we have played a lot with the sequencer. We have done, of course, application in the human genetics, microbiology. But today I would like to show you how to apply this technology to proteomics, in a sense, in order to find out the epitopes, the collection of antigenic regions in proteins that could be of interest in several different fields. In particular, I will show you the results for the celiac disease, which is just an example. What we call epitome is just a collection of epitopes that are presented and are recognized by antibodies generated by my own body. This kind of information is relevant for several diseases that are autoimmune diseases. The celiac disease is one of them. It's not one of the main important because it's pretty frequent, but fortunately there is a pretty simple way to overcome the problems. Antibody profiling in autoimmunity, as you can read there, is relevant not only for diagnostic purposes, of course, but also for studying the natural progression of immune response, for identifying the subset of patients that could share common clinical features, for prognostic purposes, to monitor responses to antigen tolerizing therapies that could be relevant in other diseases, and to discover novel autoantigens. The disease we choose, I told you, is the celiac disease, which is a complex disease in a sense because it's very simple to overcome the effect, but it is multifactorial. There is a strong association with other autoimmune diseases. This is not known, at least I think, to the general public. There is a well-known antigen, a triggering antigen, which is the gluten, which is present in the wet. So it's pretty diffuse in the normal foodstuff. The main autoantigen is transglutaminase, which is used as a marker. And not only the children are affected strongly by this disease, but also adults are coming more and more affected by this disease because they are exposed for a long time, of course, to a kind of diet that in Europe and in Italy in particular is pretty common. However, fortunately, this kind of disease can be, the effect of the disease can be overcome quite easily. It's simple, not nice, but simple to remove all the gluten from the diet and decrease the effect on the patient from the celiac disease. The approach we would like to propose, and we have already used for not just for the celiac disease, but also for the diabetes and for identifying other epitopes that are interesting in different diseases, is the combination of three different technological platforms. On one side, the phage display selection, which is pretty well known in biotechnology, 454 sequencing, of course, and at the very end, the protein microarray technology. The idea is the following. The patient in their sera contains not just the antibodies, a collection of antibodies, but also autoantibodies, the red one depicted there. Is there a pointer? Okay. Our idea was to combine the sera coming from the patient with a library, a cDNA library, so expressed transcript that have to be normalized then fragmented and cloned in a phage library. The library comes from the target organ. In this case, the target organ is the colon, the pancreas, and so on. So you can pull them, have the largest collection possible of transcript that could be related to the function of the target organ, have a phage library, the larger the better, and combining, pretty obvious, combining the antibodies with the phage that display on their surface all possible epitopes coming from the cDNA, not from the patients. I would like to stress the fact that this cDNA library is taken from different individuals, from different samples. It's not necessary to have the material coming from the very same patients. It's simply from the target organs. Combining these two materials, you can have the recognition from the autoantibodies of their proper target, the antigen. Of course, you will find out several of this kind of antigens. The very final result is to take out the information. This red fragment contains the genetic information coding for the antigen, and collect the so-called epitome, the collection of all the possible interesting autoantigen that are related to the pathology we are looking at. In the past, our colleagues in Novara, which were interested in this disease, were working this way, but using a traditional approach. After selecting the clones that were recognized by the autoantibodies, they plated the colonies, picked up a certain number, let's say 1,000, which was pretty reasonable. They grew the colonies in microtiter plates, purified the proteins generated that way, printed those fragments on a protein microarray, and then, using again the sera from the patients, tried to find out the positive clones. In this way, they were able to collect some of the epitome, a few positive clones from this very large collection of potentially useful fragments. The approach we have pursued was very similar in a sense, but much faster, because instead of plating the clones, picking them, growing them one by one, and then going to the protein microarray, we used the 454 sequencing in order to find out which was the diversity, molecular diversity within the selected autoantigens we had in the, let's say, the red family here. The approach started from the non-selected library, so the collection of the entire cDNA fragments coming from several target organs. We generated the cDNA, normalized the cDNA library, fragmented it, used this filtering or open reading frame filtering vector, which is able to take rid of all the possible fragments that are not containing open reading frames. You can imagine that fragmenting a cDNA could give you not just the proper fragments, but all the others. And this kind of vector, which has been invented by our colleagues, was able to get rid of the wrong fragments. So at the very end, we had a good library, just containing open reading frames which were related to the cDNA. Of course, the open reading frames that were coming from there started and ended in a completely random position of the cDNA, of the original transcript. This means that you are not collecting the entire protein, but just an epitope. And this is very interesting, you will see later, in terms of identification of the real fragments that are polypeptides that are interesting for the immunorecognition. After selecting sorry, this is the non-selected library. After collecting this phage library, we cut out the insert. We are not using PCR because we found out that amplifying the library this way using PCR can alter dramatically the composition in terms of number of reads per cDNA. and therefore we prefer to use restriction endonuclease to get out the inserted fragments, and then we go directly to the sequencer. The titer of the phaged library we used was 1.5 million clones, and we decided to run a sequencing in order to get at least 1/10 of the library, in order to have an idea of let's say, of the number of genes and the normalization procedure, the goodness of the normalization procedure. We set up a website that we are actually using for several different projects that is not able just to present the results, but also to process the sequences coming from the sequencer in order to organize the database and give us the final results we are expecting. In particular, you see here the number of genes that we have found out and their, let's say, their content. In terms of non-selected library, we have been able to pick up many genes, more than 1,000 genes, but you can see here the accumulation curve related to these 150,000 reads. We are not very close to the plateau. So possibly collecting many more reads, we could get up to possibly, let's say, 15,000 or slightly less. But this means that we have a good sampling of the material we have, clones we have generated. On the other side, you see also that the library is really normalized, because most of the genes are represented just by one or few reads, and very few of them, very few genes have more than 50 reads representing their content. This means that we have no strong imbalance in terms of composition. This is good, of course, for finding out the interesting epitopes within our collection. What's very important is that 97% of the insert we sequenced contained a real operating frame. This means that our selection system is really good in getting rid of the non-interesting or let's say really chimeric fragments that we can generate fragmenting the starting DNA. This work has been already published in Nucleic Acid Research last year. Then we went directly to the selection in our disease model, the celiac diseases. And as I told you before, the celiac disease is very heterogenic in terms of association with other autoimmune disorders. So you see here we have Down syndrome, autoimmune hepatitis, and so on. So a very diverse kind of patients, in a sense, that all have the celiac disease, but at the very same time they have other problems. This means that the genetic heterogeneity of the people is pretty large. This makes the situation more complex, of course. We decided to create two pools of sera. One is pool H and the other pool J, having different persons providing their sera, female and male, so together, and with different kind of associated syndromes. These pools were used, as I told you, in order to select in 3 subsequent rounds to select the content in terms of autoantigens that could be of interest to the disease itself. After each round of selection, we cut out the insert of the phages that we selected, and we went to the sequencer in order to get out the sequence information of the and therefore the epitope information of the selected clones. And at the very end, we used the website I told you before in order to analyze the data. The overall statistics is here. We collected several tens of thousands reads from each round, 1, 2, 3, and each pool, J and H, in this study. The first thing we considered and controlled is that the highly represented genes, like this MJH9, which was pretty, I think, the top scorer in the non-selected library, were depressed in their presence after 3 rounds of selection. You see that we got just 33 reads, 27 against 710. And this means that this gene, which was pretty well represented in terms of quantity during the at the very beginning, was strongly deselected after the 3rd round of selection in both pools. On the other side, we have the opposite, which is what we would like to see. Top one, which was almost present in our first analysis of the non-selected library. We got just 2 reads out of 150,000 of top one, increased strongly after the 3rd round in H and the J pool. We got 110 reads and 700 reads in the 2 different pools. This means that this, and we know already, this is one of the top scorer. In this case, the name of the gene is similar to this is rank. One of the top scorer in terms of capability of recognizing the autoantibodies coming from the pool of sera we have used. You see an overall picture showing the top scorer in J and H pool 3rd round respect to their original number of reads in the non-selected library. All these fragments were almost not present in the non-selected library, or very few reads in the non-selected library, but there was a strong increase at the very end of the selection process. This means that we are able to increase the concentration, in a sense, of the interesting epitopes using as a selector the sera coming from the patients. Using the 50 top scorer in the selected and non-selected library, we got this ranking list, and if we control let's say the overimposition of the genes, the first list against the other 2, C1 means the J pool, let's say, and C2 means the H pool. You see that there is a very low overimposition of the 3 lists. This means that the non-selected library for the 50 top scorer is really well distributed. There is a lot of genes, different genes that are not related to the disease itself. This library is a general library. The pools coming from patients with some specific genetic characteristics and the other one which has different genetic characteristics, although both are celiac. The 2 pools are closer, more similar, but you see that the genes that are in common between the first pool and the other pool are not so many. And this is interesting because this means that, as we already know, the celiac disease has a strong autoantigen, and we already know that, but the population that has the celiac disease is very different in terms of other diseases associated to this kind of problem. And this is already known, but we have a positive results in terms of this genetic variability. If we consider each pool per se, we have this kind of situation. You see that the first round of selection, 2nd round of selection, and 3rd round of selection for the J pool, and the very same for the H pool. We have a list of genes that are in common between the top scorer 50 genes in the 3 rounds of selection. If we consider their intersection, we see that the 1st, 2nd, and 3rd round for the J pool generate a list of common genes, let's say, that is equals to 15, the yellow one here depicted. For the H pool, we have a list of 25, but if you consider the 2 lists, as I told you before, if you consider the 2 lists taken one against the other, there are not so many genes that are similar one respect to the other. This means, again, is a demonstration of what I told you before, that considering these people all together, the celiac disease people, affected people, is not a good idea, because from the immunogenic point of view, these people are pretty different. You can treat them for the celiac disease, but for all the rest of problems they have, they are really different people. So we should consider that. We already knew that information, but from the immunological point of view, we have a demonstration of this. One of the very interesting features of this approach is that we are not only able to pick up a gene, a specific gene that could be related to a specific disease, but also we are able to define an epitope. If you look at the situation related to this gene has several exons depicted here along the genomic sequence, and in the different rounds of selection, H1, H2, H3, you see there is some finding here in green where we were able to find out the sequence corresponding to this coding region. There is a specific region, this one, which is here in boxed, that in all three selection processes was present, and in particular this sub-region, blue one, was the key feature in terms of anti-immunogenicity, which was able to give a good score to the gene itself. Therefore, you have not to consider the entire gene as an interesting marker, but in particular this epitope. From the possibility of generating, let's say, antibodies, monoclonal antibodies for, let's say, just diagnostic purposes, this is a key information because you are able to synthesize this peptide, which is in this region, and not the entire peptide covering the entire gene. Use just this peptide and then go directly to the production of polyclonal or monoclonal antibodies. And this is a really interesting information from the structural point of view. Again, this is an example. This was one of the common genes, let's say, between H and J pool. And you see that the tiling of sequence information we got from the phage library gave us the possibility to find out this core region, which is the one between the two arrows, let's say, that is the auto antigen that we should consider for further studies. For validating the genes that we found commonly in this selection of autoantigens, we used 46 other different patients for the celiac disease and 44 for the healthy individuals in order to create a control using a very different technique, which is not sequencing based, but a typical, let's say, ELISA or protein microarray approach. The protein microarray has been generated by our colleagues by amplifying just those genes that were in those regions that were identified by the previous screening made by next generation sequencing, and spotting the peptides generated this way by expressing them on a slide, hybridizing against all the 46 celiac disease sera, as I told you before, and the controls. And the results you see in this picture are pretty different. This is the B panel shows the results coming from 2 patients. So the results are pretty positive in a sense. On the C panel, you see the results coming from controls. Of course, controls in principle should not react against themselves because they do not, in principle, have any kind of disease. And collecting these kind of results and the results coming from ELISA test, which is the gold standard in this field, of course, we have been able to pick up the best scorer, in a sense, the best, the list of best antigens that could define the entire population of people showing the celiac disease symptoms. Again, I would like to stress the fact that all these people are different, not just because, or in principle, we are all different, but because they have almost always associated a different kind of disease with the celiac disease itself. So this list of 13 genes is the top scorer list after validation with a completely independent way of doing the experiment, proteomics in a sense. But if you look closely to results for a list of 6 of the very best genes, you see that indeed the two populations, celiac diseases and healthy individuals, are pretty different in their behavior with respect to this antigen and this one and so on. So it's pretty simple to look at them and say, okay, the two populations indeed are different. But if you consider a new sample coming to your lab, you see that there is it's not that simple with a single marker to say that one individual is positive, is a celiac disease affected individual or not, because there are many, as you can see here, there are many celiac disease individuals that are not positive for this marker, even here, even though it's true that most of the healthy individuals do not show any positive signal for these markers. So we found out that only combination of several markers all together can give you the correct picture related to the disease and to the features of the individual. And this kind of approach we are going to apply, we are already applying to very different situations. The celiac disease is pretty complex in a sense, even as I told you, it's pretty simple to treat the symptoms. But in several other occasions it's possible to subdefine the population you are looking at using this kind of markers, which are not in a sense genetic markers, but immunological markers. In conclusion, this was just a presentation related to the possibility of combining and contaminating different technologies, in particular the 454 sequencing technology, the phage display, which is at the very basis of the entire process, and the protein microarray, or ELISA if you like, which is much more expensive in a sense for testing the results themselves. What is really interesting and clever in using the sequencing technology is the extreme the increase in the speed you can reach for getting out the immunological information you want to have. Just the very last one showing you the people that are collaborating with us in this project and are collaborating with us for several other kinds of applications. Thank you very much.
Thank you very much. Very, very, very interesting. Congratulations. So, question, please.
The first one is there a functional category enrichment in the target genes to identify? And the second one is perhaps an naive observation. Would it be reasonable to use this platform to characterize the epitome, for example, in response to viral infections?
Yeah, I will express again your question. On one side, the question was, is there any functional enrichment of the genes you are selecting? I have to say that starting from a cDNA library, indeed there is a bias, because you could not be sure that the cDNA library you are starting with contains all possible epitopes that could be of interest for your specific study. And it's not just it's not related to the functional bias, but to the starting material that could not contain at that moment of sampling the transcript that you are looking at or you are interested in. The other question, the possibility of applying in infectious diseases this kind of approach, the answer is for sure yes. We are already doing pursuing this approach for pathogens that are known to be widespread in Asia in particular, and there are people that are infected. We are using their sera in order to find out which is the epitope that is recognized by the people that are firstly infected and they are rescued from infection, in order to be able to define the genes and the epitopes in particular to be used for developing a vaccine for them.
Thank you. Fulvio. Sorry.
Okay, since you mentioned the issue of where do you get the RNA from to start your approach, did you think about looking at tumor infiltrating lymphocytes and trying to characterize epitopes in that particular setting in which probably you have an enrichment in antibodies or T-cell receptors that actually recognize tumors?
We haven't done that, but of course it would be a good idea to do that. I have to say that in principle, coming back to your first question, in principle it would be possible even to use the entire human genome to generate the phage library containing all possible fragments. And using the power of the filtering system, which is based on the characteristic of the vector, it would be, in principle, possible to get out just the open reading frame. This means that not just those expressed, of course, but all present in the human genome. This would require, however, a very, very, very large library, and that was the hurdle we faced when we decided not to move in that direction. But for bacteria, we are going to use exactly that approach, because instead of 3 billion bases, we are going to play against 3 million bases, which is pretty more reasonable. So we are not going to use the transcript coming from the bacteria, but instead using directly the genome, inserting the fragments of the genome in the vector, and try to express the open reading frames which are in there to play against them.